Hello, everyone, and welcome back to the College of Glycation, a series devoted to unraveling the science of how sugar impacts our bodies through glycation. I am once again Paul Reynolds, a biomedical scientist and professor of cell biology. And today's topic is yet another compelling example regarding the subtle but powerful ways glycation can affect the human body, one organ system at a time. I'll be your host, and today's episode takes us into perhaps the most intimate and biologically consequential spaces of all, human fertility. As we've discussed in earlier episodes, glycation is the spontaneous bonding or attachment of sugars like glucose and fructose to proteins, fats, and DNA. It's a non-enzymatic reaction, meaning it happens without the body's control, and it results in a range of reactive compounds that we call AGEs, or advanced glycation end products. Once formed, AGEs can disrupt cellular function, provoke oxidative stress, trigger inflammation, and gradually damage tissues throughout the body. So today we're zooming in on how this insidious biochemical process may be disrupting something fundamental, our ability to conceive, to carry a healthy pregnancy, and to give rise to the next generation. We'll explore how glycation impacts the testes, ovaries, endometrium, and placenta, and how it relates to common reproductive disorders like polycystic ovary syndrome, or PCOS, endometriosis, male infertility, and gestational diabetes. We'll also talk about what can be done about it, importantly. So let's get to it. Let's begin by acknowledging the growing crisis in reproductive health, one that spans both men and women. Globally, infertility affects about 15% of reproductive age couples. That's roughly 1 out of every 6. Of these cases, male factors account for about 30%, female factors maybe another 30%, and the remaining are unexplained or due to a combination of both. On the female side, the most common causes of infertility include PCOS, polycystic ovary syndrome, which affects about 1 in 10 women of reproductive age. It's a metabolic and endocrine disorder characterized by irregular ovulation, excess androgens like testosterone, and multiple ovarian cysts. But it's also deeply tied to insulin resistance and metabolic function, and that's where glycemic control and glycation can take the stage. Endometriosis is another major contributor in women. It affects approximately 10% of women worldwide. In this condition, endometrial-like tissue grows outside of the uterus, causing chronic inflammation, pain, and again, often infertility. And then there's diminished ovarian reserve, which may occur earlier than expected in some women, leading to a shortened reproductive window. Meanwhile, male infertility has been quietly escalating over the past several decades. Sperm counts have dropped by more than 50% in Western countries since around the 1970s. And there are many factors that are involved, including environmental toxins, endocrine disruptors, and obesity. But mounting evidence points to a powerful metabolic component as well. So how does glycation fit into this picture? Well, let's start with male fertility. Advanced glycation end products, or AGEs, have been found to accumulate throughout the male reproductive tract, particularly in the testes, the epididymis, and sperm cells themselves. In a study published in the International Journal of Andrology, diabetic men had significantly more AGEs, and in this case, the researchers looked for CML, carboxymethyl lysine. That's a particular type of AGE. And they found that they had much more of that type of AGE on their sperm cells compared to healthy men. These glycation products were located throughout the sperm body, so throughout the entire cell, but they were most intensely focused on the acrosome. That's the very front leading edge of the sperm cell, as well as the tail. That's the flagellum that propels the cell. Those areas are both crucial for fertilization and motility. Those research endeavors have been published by the author Maladis in 2009. Now, the concern here isn't just cosmetic. That same study found that these glycated sperm cells had more DNA fragmentation, which again impairs fertilization, embryo development, and increases the risk for a miscarriage. There was a second study that reinforced these same findings, showing that diabetic men had lower sperm quality with altered testicular gene expression. And those metabolic changes were closely linked to age accumulation. Again, that research was a few years after the first paper I've referenced by Maladis and coworkers, in this case 2011. In an animal model published just last year, researchers have treated diabetic mice with a DNA aptamer, and that basically binds to ages and blocks their harmful effects, basically hiding or protecting ages from compromise. The result in this case was that sperm motility, the rate at which it can travel directionally, sperm count, and viability were all significantly improved when ages were disrupted, even without altering glucose levels. These data suggest that ages, and not just high blood sugar or hyperglycemia, are direct drivers of infertility. That was published by Dr. Mori in 2024. The general takeaway is that glycation interferes with male reproductive health at a structural and molecular level, and its effects may go unrecognized in standard fertility assessments, unfortunately. Now let's talk about female fertility, and here the story becomes even more intertwined with metabolic health. PCOS, as we mentioned earlier, is one of the most common causes of female infertility, affecting up to 20% of women in some populations. While PCOS is often framed as a hormonal disorder, many researchers now correctly recognize it as a metabolic syndrome, marked by insulin resistance, again, our body's failure to respond to the clue insulin, abdominal obesity, and in many cases, chronic inflammation. One of the key links here is the accumulation of ages, because ages are very central drivers of chronic inflammation. In a systemic review published in Human Reproduction, ages were shown to impair granulosa cell function. These are the cells that surround and nutritionally support developing eggs in the ovaries. Disrupted granulosa cells activity leads to poor follicular development, anovulation, which is a failure to ovulate, and therefore lower fertility. This was published in 2014 by Mary and colleagues. Another paper, this time from Human Reproduction in 2011, looked at levels of ages in the follicular fluid of women undergoing in vitro fertilization, or IVF. The results in this case were clearly striking. Higher age levels correlated with fewer egg cells, or oocytes, retrieved, lower fertilization rates, and reduced likelihood of ongoing pregnancy, even in younger women. These results tell us that glycation is acting independently of chronological age to compromise egg quality. And it's not just the eggs, mind you, the endometrium, that's the lining of the uterus, is also susceptible to glycation. Ages can interfere with endometrial receptivity, making it harder for embryos to implant, even after fertilization has occurred. Now let's talk about pregnancy. Pregnancy should be a protected window, but in a high glycation environment, it can go wrong in subtle and not so subtle ways. Gestational diabetes is something we should cover first. This is a condition in which a woman without diabetes develops high blood glucose during pregnancy. Gestational diabetes affects about 6 to 10% or so of pregnancies in the United States annually. It's associated with increased risk for preeclampsia, premature birth, and larger than average babies. But here's the kicker. Ages may play a central role in such placental dysfunction. One review showed that ages can impair placental development, promote inflammation, and reduce nutrient exchange to the fetus. That was published by Dr. Zhu in 2020. This not only endangers the pregnancy, but may also program the child for future metabolic issues, putting the child in a compromised position going forward. So, what do we do about it? Well, let's shift now to some solutions. This is where the picture shifts and becomes hopeful. First off, ages are modifiable. Yes, some glycation is inevitable with age, but the rate of accumulation is directly tied to our metabolic state and lifestyle. One powerful lever to wield in your defense is your diet. High temperature cooking methods like excessive grilling, frying, and roasting produce larger than normal amounts of dietary ages. These are the exogenous ages we talked about in the most recent episode. Lowering your intake of ultra processed, highly browned, and caramelized food can reduce age levels in your blood and tissues. Another lever that can be used is exercise, particularly resistance training, that's key, and high intensity interval training or HIIT exercises. Both of these are proven to improve insulin sensitivity, and therefore it reduces oxidative stress and slows age formation. If you are more sensitive to insulin, you'll more effectively package glucose away in a protective way, avoiding excess glycation. And then there are promising therapeutics. In a randomized controlled trial, women that are undergoing in vitro fertilization were given an extract of Trapa bispinosa. That is a traditional herbal medicine known to block age formation. And those individuals had a threefold higher chance of live birth. Not only did they have lower age levels in their blood and follicular fluid, but their oocytes developed better and their endometrial receptivity, again, the likelihood of an effective implantation improved. These results suggest a new frontier in reproductive medicine, one that addresses biochemistry at the source rather than just simply treating symptoms. Much more research is needed. But let me leave you with this today. Fertility, as you may now know, is not simply about hormones and genetics. It is a metabolic state, a reflection of whole body health, from the bloodstream to the nucleus of individual cells. Glycation, while subtle and slow, acts like rust on the machinery of reproduction, affecting sperm DNA, egg viability, placental function, and embryo development. But by understanding glycation, we empower ourselves and others to take control of a system that too often feels out of control. Thank you, one and all, for joining me today in this episode of the College of Glycation. If you know someone facing fertility struggles, whether it's a couple trying to conceive or a practitioner looking for deeper root causes, consider the new information you learned about today. The evidence from peer-reviewed studies backs up the concept that our diet fuels reproductive compromise. When we understand how glycation damages tissues, we can take steps to interrupt that process and rebuild. Thanks again for listening, everyone. Until next time, remember to stay curious and stay metabolically healthy.